Hello, welcome to World of Microbes. The topic of today's video is nucleic acid. Every physical trait, every biological process, and every living organism on this planet relies on a single, microscopic powerhouse, the nucleic acid. Without them, life as we know it simply wouldn't exist. Welcome to the invisible world of DNA and RNA, the ultimate instruction manuals of the universe. Let's dive in. Nucleic acids are large biomolecules that play essential roles in all cells and viruses. A major function of nucleic acids involves the storage and expression of genomic information. Deoxyribonucleic acid, or DNA, encodes the information cells need to make proteins. A related type of nucleic acid, called ribonucleic acid, RNA, comes in different molecular forms that play multiple cellular roles, including protein synthesis. Nucleic acids are long-chain polymeric molecules. The monomer or the repeating unit is known as the nucleotides, and hence sometimes nucleic acids are referred to as polynucleotides. Nucleic acids can be defined as organic molecules present in living cells. It plays a key factor in transferring genetic information from one generation to the next. Nucleic acids are composed of DNA deoxyribonucleic acid and RNA ribonucleic acid that form the polymers of nucleotides. Nucleic acids are polynucleotides, that is, long-chain-like molecules composed of a series of nearly identical building blocks called nucleotides. Each nucleotide consists of a nitrogen-containing aromatic base attached to a pentose, 5-carbon, sugar, which is in turn attached to a phosphate group. Each nucleic acid contains 4 of 5 possible nitrogen-containing bases: adenine, A, guanine, G, cytosine, C, thymine, T, and uracil, U. A and G are categorized as purines, and C, T, and U are collectively called pyrimidines. All nucleic acids contain the bases A, C, and G. T, however, is found only in DNA, while U is found in RNA. Ever wonder what actually makes you, you? It all comes down to DNA. Think of DNA as a massive instruction manual written in code. It looks like a twisted ladder. The blocks that build it are called nucleotides. Every single block has 3 parts: a sugar, a phosphate, and a code letter. There are 4 letters: A, T, C, and G. Here the order of these letters spells out your traits. But they have strict rules. A only pairs with T, and C only pairs with G, and millions of these pairs create the unique blueprint for your entire body. Think of nucleotides as the ultimate molecular Lego bricks. When you snap them together, they build your entire genetic blueprint, DNA and RNA. Every single nucleotide has 3 simple parts. First, a sugar base. Second, a phosphate connector. And third, a nitrogen base, which acts like a letter in a secret code. In DNA, those code letters are A, T, C, and G. They pair up perfectly to form that famous twisting ladder. In RNA, the T is swapped for a U, taking instructions out to build proteins that keep you alive. But nucleotides aren't just data storage. One special, souped-up nucleotide called ATP is your body's personal cellular battery. It provides the pure energy for every breath you take and every move you make. From coding your eye color to powering your brain, nucleotides are the tiny engines driving life itself. The pentose sugar in DNA, 2-deoxyribose, differs from the sugar in RNA, ribose, by the absence of a hydroxyl group, OH, on the 2' carbon of the sugar ring. Without an attached phosphate group, the sugar attached to one of the bases is known as a nucleoside. The phosphate group connects successive sugar residues by bridging the 5-hydroxyl group on one sugar to the 3-hydroxyl group of the next sugar in the chain. These nucleoside linkages are called phosphodiester bonds and are the same in RNA and DNA. Ever wonder how your body stores life's entire instruction manual? It all comes down to just 5 chemical letters called nitrogenous bases. These are the building blocks of your DNA and RNA, and they are split into 2 exclusive clubs. Let's break them down. First up, meet the purines. Think of them as the heavyweight champions. They have a double ring structure. The members? adenine and guanine. A quick memory trick to remember this, always good. Pure gold, double the rings. Next, we have the pyrimidines. Don't let the big name fool you, these only have a single ring. This club includes cytosine, thymine, and uracil. To remember these, just picture cutting a single slice of pie, CUT. But here is the catch, they can't just mix with anyone. They follow strict dating rules called complementary pairing. In DNA, A always bonds with T, and G always bonds with C. If you are looking at RNA, thymine takes a break, and uracil steps in to pair with A. These simple pairs create the entire code for every living thing on Earth. Pretty wild, right? Meet the phosphate group. It is a critical component of every single nucleotide, the building blocks of DNA and RNA. Structurally, it is simple, just one phosphorus atom bonded to 4 oxygen atoms. In a nucleotide, this group hooks onto the 5' carbon of the pentose sugar. But its real magic happens when nucleotides join together. The phosphate of one nucleotide chains up with the next nucleotide sugar. This linkage is called a phosphodiester bond. It forms the incredibly strong, repeating sugar-phosphate backbone that protects our genetic code. Because of its oxygen atoms, the phosphate group carries a strong negative charge. This makes DNA highly water-soluble and allows it to interact perfectly with cellular proteins. Plus, splitting these phosphate bonds releases the raw energy needed to build genetic material in the first place. So, the next time you think of DNA, remember the phosphate group, the negative powerhouse holding life's blueprint together. Now let's break down the structural difference between a nucleoside and a nucleotide so you never mix them up again. Let's start with the basic building block, a nucleoside. A nucleoside is made of just 2 parts. First, you have a 5-carbon pentose sugar, which is ribose in RNA or deoxyribose in DNA. Attached to this sugar at the number 1 carbon is a nitrogenous base. This base can be a purine, like adenine or guanine, or a pyrimidine, like cytosine, thymine, or uracil. When you have just the sugar and the base joined together, you have a nucleoside. For example, adenine plus ribose gives you the nucleoside adenosine. Now, how do we get a nucleotide? It's simple, you add a phosphate. When a cell takes that nucleoside and uses a kinase enzyme to attach 1, 2, or 3 phosphate groups to the sugar's number 5 carbon, it becomes a nucleotide. Think of it as an algebraic equation, nucleoside plus phosphate group equals nucleotide. Because of that added phosphate, nucleotides are highly acidic and carry a negative charge, which gives DNA and RNA their overall negative charge. Classic examples of nucleotides include adenosine monophosphate, AMP, adenosine triphosphate, ATP, your cell's energy currency, and the individual links that string together to form DNA and RNA chains. To wrap it up, remember that S in nucleoside stands for sugar plus base, while the T in nucleotide stands for 3 parts, sugar, base, and phosphate. Deep inside every living cell on Earth, a microscopic library holds the ultimate software of life. This software is written in the language of nucleic acids. While there are millions of different proteins and structures that build a living organism, they are all governed by just 2 master molecules, deoxyribonucleic acid, or DNA, and ribonucleic acid, known as RNA. Before we look at how they differ, we need to look at what they share. Both DNA and RNA are built from the exact same structural blocks called nucleotides. Think of these as molecular Lego bricks. Every single nucleotide contains a phosphate group, a 5-carbon pentose sugar, and a nitrogenous base. It is the sequence of these bases that actually holds the code of life. Let's start with the master blueprint, DNA. DNA is famously shaped like a double helix, a twisting ladder. It consists of 2 long strands running in opposite directions, locked together tightly. Chemically, its name tells its story. Deoxyribose means its 5-carbon sugar is missing 1 oxygen atom at the 2' position. This tiny omission is actually a massive evolutionary advantage. Without that oxygen, DNA becomes incredibly stable, resistant to chemical attacks, and capable of lasting for thousands of years. To store information, DNA uses for chemical letters or nitrogenous bases, adenine, thymine, cytosine, and guanine. Adenine always pairs with thymine, and cytosine always pairs with guanine. Protected safely inside the nucleus of your cells, DNA acts as the permanent, unmoving hard drive of an organism, preserving genetic data across generations. But a blueprint is useless if you can't read it. That is where RNA comes in. Unlike its rigid, double-stranded sibling, RNA is generally single-stranded. This makes it highly flexible, allowing it to fold into complex, three-dimensional shapes to perform physical work. Structurally, RNA uses a sugar called ribose, which keeps that extra oxygen atom. This makes RNA highly reactive and temporary. It is built to be used quickly and then broken down by the cell. RNA also changes the alphabet slightly. It uses adenine, cytosine, and guanine, but it completely replaces thymine with a base called uracil. So, in the world of RNA, adenine pairs with uracil. To see them work together, think of a secure kitchen. DNA is the priceless, heavy cookbook that can never leave the library. If you want to bake a protein cake, you can't take the book. Instead, you make a temporary photocopy. This photocopy is messenger RNA, or mRNA. It carries the recipe out of the nucleus to the cell's factory floor. There, ribosomal RNA, or rRNA, forms the physical factory machinery itself. Finally, transfer RNA, or tRNA, acts like a delivery truck, bringing the exact amino acid ingredients required to build the final protein chain. In short, DNA is the architect, holding the permanent, stable master plan. RNA is the construction crew, taking temporary copies of those plans to actively build the organism. Together, they form the unbreakable foundation of all life on Earth. Nucleic acid is responsible for the synthesis of protein in our body. RNA is a vital component of protein synthesis. Loss of DNA content is linked to many diseases. DNA is an essential component required for transferring genes from parents to offspring. All the information of a cell is stored in DNA. DNA fingerprinting is a method used by forensic experts to determine paternity. It is also used for the identification of criminals. It has also played a major role in studies regarding biological evolution and genetics. That brings us to the end of our breakdown of the two master nucleic acids. Now you know exactly how DNA and RNA are built, and how they share the massive responsibility of managing the code of life. But knowing the structure is just the first step. How does DNA actually copy itself? And how exactly does RNA turn that code into physical proteins? We will dive deep into the fascinating machinery of replication, transcription, and translation in our very next video. You won't want to miss how this molecular factory works in real time. Make sure to hit that subscribe button, turn on notifications, and stay tuned. Thank you for watching, and we'll see you in the next video.